So Woody was a graduate student with you, and what was it that you and he did together that was most noteworthy?
Well, Woody was part of a program we had at Stanford where people earned, I think, a Ph.D. and an M.D. simultaneously, and he was part of that program. The work that he did a number of really very good experiments while he was with me, and the one that's noteworthy in respect to aging and our efforts to understand this cell memory phenomenon, which incidentally should not be called a clock, but what I called later what was the name I gave?
A replicometer?
Sorry?
Replicometer.
A replicometer, yeah. Because it's not a clock, it doesn't measure the passage of time, it measures the number, actually, as we learned later, initially we thought it measured the number of cell replications, but it actually technically measures the number of DNA replications. But Woody and I decided that we have to go forward and find out, first of all, where this replicometer is located. Is it in the cytoplasm or is it in the nucleus? So Woody did a very interesting experiment based on the technology that we could use, in which you could separate nuclei from cells and then fuse nuclei back into cells. And having that technology available, you could now take the nuclei from young cells, that is those cells early in the population doubling sequence, and put them into the cytoplasm of cells that are later in the sequence, and the converse. That experiment was done by Woody, and it was also done indirectly in another way by Muggleton Harris, a woman who worked in my lab about the same time. Both studies revealed that the replicometer was located in the nucleus, which was not a major discovery in the sense that one could probably guess that's where it was. But at least it provided good evidence that that's where the replicometer was. And then Woody, I think, left for another job at the University of Texas. Things were slow to develop, at least in my lab at that time. We didn't know where to go with this question until a major incident occurred. And as is not uncommon in science, it was an accident. It was an accident in which a lady by the name of Carol Greider, decided to visit her boyfriend at McMaster University in Canada, where Calvin Harley, a former, I think I can safely say, scientific grandson of mine, was working, and he was working with my system of finite growth replication.
Cal had studied with Sam Goldstein again, small circle of researchers in this field.
Exactly. And Cal was introduced to Carol. She was a visiting scientist, actually. She was working with telomeres in a protozoan, and knew a lot about telomere biology. Calvin knew very little, and Carol knew very little about my system, if anything, and was introduced to it by Cal in a discussion. And they decided, as a result of that discussion, to investigate the behavior of telomeres in my system. And sure enough, they made a major discovery, that during the course of the replicative limitation in normal human cells, the telomeres became shorter and shorter and shorter. Telomeres, for those who are not familiar with the biology, are structures at the ends of chromosomes containing repeated sequences that don't provide genetic information, but as we learned later, are a kind of buffer system that protects the information-containing genes downstream, because the replication of DNA does not permit the replication of the portions of the chromosome at the very ends. So if you provide the chromosome with nonsense material at the very ends, and that gets eliminated as a result of normal DNA replication, then you don't damage downstream genes, which of course you don't want to do. So it was discovered that telomeres become shorter and shorter during the period of the replicative senescence of normal human cells. Well, that was a major discovery.
Well, let's back up, though, a few more years before that. So I once had the pleasure of talking to Alexei Lovnikov, a Russian theoretician. Alexei tells the story about how he was in Moscow listening to a lecture about Dr. Leonard Hayflick's discovery that human cells age. And he says it was a fall day, and he was watching the leaves fall from the tree. Came to a subway station there in Moscow and had this flash of insight that he knew exactly how this clock worked, and that it was the telomere clock. So he laid out the theoretical model.
Yeah.
I was studying, when I entered the field with Sam Goldstein, Sam had an instinct, I can't explain it, that there was a change in the structure of repetitive sequences in DNA. You remember he was studying the inter-allele repeats and all these allele repeats and so on. And it was in Sam's lab that it was discussed, well, there's this wild-eyed, crazy idea of Alexei Lovnikov, but no one could test it. And it was only after the telomeric sequence was identified that experiment that you could describe could be performed. What's your memory of Alexei? He was obviously a Russian theoretical biologist. Did you meet him at any meetings? Did he come up to you and say, Dr. Hayflick, I have this... He wouldn't have said a wild-eyed idea about... He would have said, I know what causes, Alexei, very certain of himself. Do you have any memories of...
Well, as a matter of fact, I think I met him for the first time at a conference that you either organized or chaired. It was organized by a scientific journal and held here in San Francisco, I believe.
One of our early telomere meetings.
Yes, and that I believe was the first time I met him. But let me back up a little bit and enlarge a bit on what you described as his sudden realization of what might be the explanation for telomere attrition. My understanding is that after the meeting in which he heard a discussion of my work, he entered the Moscow subway, as you indicated, and while standing on the platform waiting for the train to arrive, he imagined that the tracks were similar to a DNA molecule. And then he reasoned that if the engine was incapable, when it came to the station, of replicating what was under the engine, but only replicating what was under the rest of the cars, that would provide a basis for attrition occurring on this long railroad track, which would be analogous to what would be happening with a DNA molecule, which I found to be a rather remarkable insight. It turned out that he was absolutely on target. When the
amazingly accurate.
Yeah.
Not only did he predict that the clocking mechanism that you, as you say, you freeze cells, they're at doubling 57, you thaw them, they're at doubling 57. Not only did he provide a molecular explanation of how that clock could work, and it being in the nucleus, as you said you and Woody had reported on, but he also proposed that it's the mechanism by which cancer cells can regain the ability to replicate beyond what we call the Hayflick limit. And that he said that there would be a special enzyme that could create this, he didn't call it the telomeric repeats, he called it the theory of marginotomy. But he had it all right from beginning to end.
Absolutely, absolutely. It's remarkable.
Another name that deserves some credit here too is Howard Cook. I don't know if you remember, but Howard
yes.
published that telomeres were shortening in human somatic cells, body cells,
Yes.
as humans aged.
Yes.
Not in vitro, not in the dish.
Correct.
Actually in humans.
Correct.
I want to say it was quite a few years before Carol and Cal's paper.
Yes, yes.
So a lot of, it shows you the interplay of theoretical scientists who don't have access to a laboratory and those that do. Ultimately, of course, it was the cloning of telomerase that allowed us the testing of that. And then, of course, there's the fun story. Do you remember you were at Geron visiting, and as I recall, filming a documentary, kind of like what we're doing right here, right now.
Yes, there was.
And walked into my office with a piece of your skin, and do you remember that day?
I was being interviewed by a TV crew, and a producer who was questioning me about my work, and he.
You came into my office asking me if I had a scalpel.
Yeah, well, that was after I replied to his question, in which he asked, well, is it true that when you take tissue from humans, all tissue from humans, it will show a limited capacity to replicate? And I said, yes. I said, and I'll illustrate it for you if you want, because it's very easy to do. How do you do that? He said. I said, well, you simply pinch your skin somewhere where you can afford to lose a little bit of it until it becomes numb, and then you pull a hair up, creating a pyramid of skin, and then you whack the top of it off with sterile scalpels. I said, do you want me to show you how to do it? He said, yes, of course, we'll film it. And as he filmed it, you walked in.
I remember you asking me for a scalpel, and I said, well, what do you have in mind? You explained this, and I said, but Len, I don't have any anesthetic. And you said, oh, damn the anesthetic.
So I anesthetized my skin by pinching it.
Okay.
It's a cheap anesthesia.
So my memory was it was mostly as a lark, as a joke. I thought, well, we always talk about the Hayflick limit. The how many doubling cells will go before they age in the laboratory dish. I thought, well, let's measure the true Hayflick limit, the number of doublings that your cells would go.
I remember that. Yeah.
But that was the joke. But just as it turned out, coincidentally, just at about that time, we had finally, after some $30 million of investment, finally had found the telomerase gene. My wife had cloned the gene. And I thought, seemed perfectly fitting that your cells would be the first ever human cells to be transfected, immortalized with the test. Of course, it could have been the greatest failure of all times. What would be the chance? What would you have guessed? My wife said, quite candidly to me, when pigs fly. All this work. What would be your guess that something...
She and Khrushchev, it's Khrushchev who made that remark, or when shrimp learned to whistle, I believe, is what he said.
Shrimp learned to whistle, I hadn't heard that one. What would you have guessed, though, would be the probability that experiment would have worked prior to seeing the outcome? It seemed highly unlikely.
It's about as close to zero as you can get.
I remember, wasn't it here on the coast near your home? Was it, do you remember we were out up for a restaurant when I told you about...
Yes, about Geron.
What restaurant was that? I don't remember.
It was at the Sea Ranch Lodge restaurant, still operating.
So it turned out to be, at least in large part, most of what we call cellular aging is this telomere replicometer ticking away. We knew that most tumor cell lines would replicate indefinitely, as opposed to normal somatic diploid cells. I called Vince Cristofalo, your old colleague, at the Wistar Institute, and I asked him, and I didn't think of calling you, Vince, because he had grown several osteosarcoma lines from cancer, and I asked him, did these cells ever slow down after you'd start to culture them? Or did they look immortal from day one and just continue to proliferate? And he said, well, they always just seem to grow linearly. And what I was getting at, and what he was answering was, do cells immortalize in people in the process of becoming cancer, or is this all, something that's generated in the laboratory? And when Vince seemed quite convinced that malignant tumor cells are immortal once put in the laboratory dish, that we organized this really large effort to try to identify how many different kinds of cancer had immortalized that way. And it turned out over 95% of all cancers.
Yeah, well, that, as I understand it, even to this day, that the most significant distinguishing characteristic between normal cells and cancer cells is the elaboration or production of telomerase by cancer cells.
There's
expression of telomerase.
Yeah. Some cells can immortalize by an alternative mechanism,
Yes.
but it also maintains the telomere. In doing that.
Yes. And
so,
I think we also now know that certain blast cell populations, gut epithelium, gut cells, blood-forming elements, and epidermal cells produce telomerase in detectable amounts.
Yeah. The telomerase was cloned from an inflamed tonsil, in fact. So in recent years, we've seen, the discovery of how this mechanism works, indeed proving it by being able to immortalize cells. But a lot of other significant advancements in medical research, the sequencing of the genome, next generation sequencing technology, where we can now sequence your DNA for $1,000 in same day, as remarkable as that is. I guess my last question for you is, for all those watching us in the future, is, what can we learn from the lessons of the past and applying it to what we believe is going to be the really serious strain on our healthcare system due to the aging of our baby boom population? There's a lot of concern about how this is going to economically stress the United States. For some of us, I think like you and I, there was a concern about how this is going to impact people on an individual level and families, But what can we learn about the nature of scientific revolutions, about so often how people are skeptical about the role of science in society, and how we can chart a better course in the future for how science is organized nationally or on an individual level to solve some of the problems of human aging? What are we doing right or doing wrong, and how can we get it better in the future, in your mind?
Well, I have several thoughts in response to that observation. First of all, I think it's important to emphasize that my experience in having at least 10 years of being ridiculed and not believed in respect to the experiments that we described is an important aspect of science, that criticism of observations is necessary in order to provide truth to what observations may have allegedly been made. So I am not at all concerned about the 10 years that I spent being ridiculed and not believed until the events ultimately were. But getting back to the main thrust of your observations is that the field of aging is, despite what we may read, has been seriously neglected in respect to an understanding of the fundamental biology of aging. This failure is based on a number of causes, one of which is the erroneous belief that the study, or even the cure, if I can use that word, or resolution of age-associated diseases, like the leading causes of death, cardiovascular disease, stroke, and cancer, that the further understanding of those pathologies will increase our understanding of aging. The answer is that they will not, that none of that research will. And I believe that this is a fundamental, what I have called, or have written about in several papers, that I've called the $1 billion misunderstanding. Because the amount of money spent on research in the fundamental biology of aging is microscopic compared to the money spent on age-associated diseases. I'm not arguing that we don't do research on those diseases. What I'm arguing is that because they're age-associated, it must be true that old cells provide the milieu, or the conditions, or the soil for the diseases that are associated with the aging phenomenon. So that an effort to understand the aging phenomenon might provide us with knowledge about the status of molecules in old cells that provide the opportunities for age-associated diseases to be expressed. There's a complete failure to focus on that idea, to say nothing of an almost complete failure to focus on trying to understand the fundamental biology of aging. This despite the fact, and this is a fact that I think is indisputable, that virtually every geriatrician, virtually every physician, virtually every scientist who has some thoughts on this process will state the following, either in the introduction of a lecture they might give or in a paper they might write, that the leading risk factor for cardiovascular disease, cancer, or stroke is aging. Well, it doesn't take more than a 13 or 14-year-old education to ask the next question: why are we then not studying the fundamental biology of aging? And the answer is we're not. I cannot understand why this apparent discrepancy exists, despite the fact that I have been questioning this for the past 20 years or more. There are many other fundamental aspects in this field. If you look at the expenditures of the National Institute on Aging, which one would think would focus on aging, is not focused on aging at all. The Institute is focused on age-associated diseases, the resolution of which will not advance our understanding of the fundamental biology of aging, just as the resolution of the diseases of childhood, like iron deficiency anemia, Wilms tumors, polio, you could name some more, that the resolution of those diseases didn't provide any insights into childhood development. They're two completely different classes of biological areas. So it's interesting, and I've done some calculations in this respect, that more than half of the budget of the National Institute on Aging, even today, is spent on Alzheimer's disease research. Only about
a very important disease, of course.
Well, there's no question about the need to do research in that area, but the resolution of Alzheimer's disease as a cause of death, and it's questionable how many cases of Alzheimer's disease actually are the cause of death. The resolution of Alzheimer's disease as a cause of death will add about 19 days on the human life expectation, which doesn't argue against doing research on Alzheimer's, but it gives you some idea of the misplacement of focus. The furtherance of that belief is that about 2 or 3%, if you are liberal in your analyses, of the budget of the National Institute on Aging is spent on fundamental biology of aging research.
Which would surprise a lot of people, I'm sure.
It will surprise many people, and I have observed that personally, and I think there's a good reason for it. The reason is that decision makers and policy makers can easily understand the pain and the suffering created by age-associated diseases that I've mentioned. But they cannot understand and don't wish to understand the fundamental biology of aging. That's very difficult for them to understand, despite the fact that they do understand, having had experience either in themselves or in associates and the family of age-associated diseases. But they have not made the connection between the fact that those diseases are age-associated, so why not study aging?
Don't you think part of it, too, is the belief that all that science and technology could potentially do is make yet one more expensive new medical modality? And what puzzles me is that when I grew up, and many of the years you were doing the research that you described here today, back in the 60s was the space race, the Sputnik era. Science was held in great esteem, and scientists were looked to provide answers. And yet today I don't see that same respect for science. Perhaps one of the last things that policymakers today would think of is of calling you and saying, Len, could science and technology potentially help us address the rising costs of healthcare in the United States rather than add to the burden of those healthcare costs? I suspect you haven't gotten that phone call recently.
No, I haven't, unfortunately. No, I haven't. But
do you share with me, though? I'm perhaps the eternal optimist, but I think that perhaps belatedly, maybe too late for this tsunami of healthcare costs coming to our shores in the United States. But nevertheless, the day will come when science and technology will make additional new breakthroughs, such as yours in understanding how cells age, the telomere story, and so on. We'll gain new insights into the biology of aging that can not only improve the quality of life for people, but reduce healthcare expenditures and make ease the burden of aging. Is that a reasonable future, do you think, if we
Well, it's reasonable only if you believe you can convert the mentality of the arch conservatives who believe that studying aging is either impractical or will have no bearing on the age-associated diseases that we're all concerned with, and which are the cause of the enormous expenditures that occur to treat clinically people in old age who usually have more than one pathology. There is, as I mentioned earlier, an almost complete failure to underwrite or encourage the study of the fundamental biology of aging, which seems to me to be an enormous blunder in respect to how policymakers are treating our health problems, most of which are associated with age.
Going back in time, earlier in our discussion, you described the day that you received that letter from the esteemed Dr. Peyton Rouse from the Rockefeller Institute saying, your conclusions about aging appear notably rash. There are people watching us that will enter the field of aging research in future generations. Perhaps they will push through those barriers, that resistance to change, resistance to the idea that mankind can, through their own efforts and through science and technology, uncover molecular mechanisms, design new therapies, and do what the current policymakers and scientists believe can't be done. Help us age better. That's my hope for future generations. Yeah. I'm hoping that they'll learn from lessons in the past, from the courage that you've displayed. And I want to thank you for that, by the way, for many years of fighting the battle. And hopefully make a better world for future generations.
Well, there's also a problem with the term aging research or research on aging. That word aging in that context could include anything associated with people getting older or animals getting older, but in particular with people getting older. It includes virtually every human institution. You can study aging in respect to its economics, to the design, to architecture, design of homes, design of nursing homes, to politics, to biology, to care, to disease, virtually everything. And that's the problem, another major problem in this field. The failure to distinguish between all of the aspects of aging, so that when you read that X million dollars is being spent on aging research, what is meant by that? If it's to cure age-associated diseases, that's not helpful. It is helpful if you're interested in the fundamental biology of aging, which you should be interested in, and then you learn, as I have and have discussed here, that very little attention is paid to that most important aspect of aging research. It should, we have in recent years used the term biogerontology, which means the study by gerontologists of the biology of the field. But that's rarely used and understood by policymakers. There is so much to criticize over the years. The field of aging has been exploited and funded to the benefit of people who generally have either no interest in or are not actually doing research in the fundamental biology of aging. It's mostly disease-oriented research or descriptive research, describing what events take place during the process of aging. Well, that's perhaps useful, but what's more useful is to understand the fundamental biology of aging, which in my judgment and in recent years has developed into an understanding that the causes are associated with laws of physics, because the cause of aging is universal, not only in biological material, but in inanimate objects. Everything that we and our listeners and viewers can see from where they're sitting or standing will age. The only difference is the time frame, how long it takes for the events to occur. The automobile that you bought new and have sitting in your garage unused for 25 years is going to age, despite the fact that it's not used. So the fundamental cause, we now believe, is associated with what's called the second law of thermodynamics. That is, the fact that energy tends to spread unless it's constrained, which means in biological terms that molecules are over time going to lose their chemical bonds and other bonding principles that hold complex molecules together. And despite the fact that we have an enormous capacity for repair, both at the DNA level and at other levels, those repair shops also undergo the same loss of energy that results in dysfunctional molecules, as do the molecules on which the repair shops work. This is now what I and a number of other people are beginning to believe is the fundamental cause of age changes.
You mentioned thermodynamics. Reminds me of the Carnot cycle and steam production and where all that field began. And it reminds me then of, as we were talking about attacking the symptoms of aging or the diseases of aging and not the core biology of aging itself. Reminds me of my mental image is a burning building. I guess maybe that's the thermodynamic part. And our loved ones outside, reaching outside the window, crying for help. And us forming a rescue operation, maybe a municipality, a fire department saying, okay, so fellow firemen, what are we going to do? Well, those boots are not optimum to go in there and rescue that girl. We need to sit down and redesign our boots. I remember a grant application, as I was looking at NIA, National Institute on Aging grant applications, it was titled "The Effect of Sole Size, the sole of your shoe, on Balance in the Elderly." And it was like a $200,000 grant to study that. Well, you could say that's a noble thing. You want people to not fall down and break a bone. But then you look at that fireman that needs to rush in and save that life, the life of a loved one that's suffering. It seems to me that the urgency that the United States is facing with the aging population, regardless of the boom, right, far should outweigh that the need to understand and the biology of aging and help our fellow human being who are not aging optimally is far more important, don't you think, than decide 90, as you say, 98% of the money goes to studying the effect of the shoes on balance in the elderly.
Yeah.
I hope that your ringing the fire bell here will maybe help make that happen and happen more quickly than it would otherwise.
Well, it reminds me very much analogously with what was occurring during the years that the poliomyelitis epidemic was prominent in the minds of people, where there were people who, stricken with the disease, were being helped by being imprisoned, actually, into iron lung machines.
Right.
And the focus on that was fine as far as helping ill people was concerned, but the real breakthrough came when attention was paid to the fundamental cause of the poliomyelitis epidemic and the development of vaccines to prevent it.
So all that technology developing the iron lung became...
it became useless and unnecessary. Well, that's where we are in the field of aging research today, in my judgment.
What if policymakers said, we don't want to spend more money studying the biology of polio because it's just going to make more expensive iron lung machines? I think that's how they think of aging today.
Because the manufacturers of the machines don't want to lose. Don't want to lose their
How much
customer
does it cost
base.
us today that smallpox was eliminated from the face of the earth? It doesn't cost us anything.
Yeah, of course. Yeah, well, I'm afraid that the policymakers haven't reached the point of understanding in respect to what we think should be done.
Yeah, I want to thank you for all the years that you put into all this field, the facing all the criticism, and thank you for reminding all of our viewers that a brighter future is possible for people like you joining the field and pushing the ball forward.
Well, thank you very much, Mike, for your friendship and your support. I really appreciate it over the years.
Thank you.